Hi everyone, welcome back to my channel. So today we are going to discuss about law of dominance, which is part of the topic in genetics. So before we get into the law of dominance, we have to understand something, a few basics in the genetics. So that are first, let us see these terminologies. F1 generation. F1 generation is what Fleal generation 1. When we take two characters, I will show it on the board. When we take two characters and then cross them, the progeny which is obtained first progeny is called F1 generation. That F1 generation is when again crossed, then you get the F2 generation. Okay, these two I will be showing in when we do the crosses. Okay, next coming to true breeding. True breeding in the sense the two gametes are pure, means the two gametes are same, like maybe tall and tall, maybe dwarf and maybe dwarf and dwarf, like that. They are true breeding in the sense that truly the same genes are present in the gamete. Okay, next coming to crossing of parents. Two gametes we take and then we cross them. Okay, when we cross them we get these F1 and F2 generations. Next, dominant in the sense this dominant gene dominates the recessive gene. Okay, dominant gene, nothing but which dominates the recessive gene is called the dominant gene. Next, coming to recessive gene, the gene which is getting dominated by the dominant gene is recessive gene. Very simple. Okay, gametes. We know very well that gametes is 23 out of 23 pairs. 22 are autosomes, one is allosome. Okay, that's very clear. The next thing that we will be seeing here is the characters. So Mendel, we know very well George Mendel. Yes, he's a dentist. He has taken 14 contrasting characters or 7 pairs of contrasting characters. Okay, let us see. Characters, dominant character, that's character. Dominant character means we have pod color, seed shape, flower position, flower color, pod shape, stem height, seed color. Now, Mendel has taken Pisum sativa, also called as pea plant, which belongs to Fabaceae family member. He has taken this pea plant, okay, as the experimental, model, and in that pea plant, he got these characters. Okay, if you observe, these are 14 characters, or you can say 7 pairs. Okay, one pair, two pair, three pair, like that. Okay, so if you observe, pod color dominant is green, recessive is yellow. Okay, in the same way, she shaped our position. All these characters are given. Now, if you observe here in brackets, I have written something: capital G, capital R, small r. Okay, these are nothing but the gamete representation. Okay, whenever we do crosses, we take these as a representation. For crossing over. Okay, now you may get a doubt here that green is given capital G, then yellow is given small g. Why can't we get yellow? Why for that? If you take y for this, when we are crossing, suppose I'm taking capital G here and y here, we can't get the cross. We get gy crosses, which complicates the cross. Okay, that's why we take capital G for dominant, small g for recessive. Okay, so this is few basics that one must understand before we learn about law of dominance. Okay. Okay, now after clearing these basic stuff, we will enter into the actual topic, that is law of dominance. Okay, so I am taking here two characters. I am taking most generally a frequently taken characters, that is tall and dwarf. Okay, so in the table that I have given just before, tall is represented as capital T, dwarf is represented as small t. Okay, now I am taking two plants. Okay, two plants I am taking. One plant is having a genotypic character of capital T, capital T. Now What do you mean by genotypic and phenotypic? Okay, two terminologies: phenotypic, okay, genotypic. Phenotypic in the sense physical appearance. Now, how is he physically? Now, suppose we take a plant. The plant is physically tall, but genetically it may not be capital T only. So genotypic means genetical combination. Okay, tall, whatever I have written tall, this tall is phenotypic character, while this capital T, whatever I have written, and here small t, whatever I have written, is the genotypic, you know, combination. Okay, these two terms are clear now. Now, these two characters, true breeding characters, these are. Now, just before I said no, true breeding means both are having same. So, same capital T, small t. Now, there is a cross in between them, maybe self pollination, or maybe not self pollination, cross pollination. Yes. Now, capital T, small t, capital T crossed with small t, capital T crossed with small t. You get capital T, capital T, small t. Now, this, whatever you have obtained, is called F1 generation. Okay. Just now I said F1 generation means filial one generation. That is when we are crossing true breeding characters. The progeny which you obtain is nothing but called as F1 generation. Okay. Now next, when you cross, suppose I am taking these two characters now. F1 characters I am taking. Okay. Capital T, capital T, small t. Now, when these two are crossed, capital T, small t, capital T, you know, small t, capital T. Yes. So you get TT, 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 TT. Yes. Now, this is looking too clumsy for us, I don't know. So crossing. For this, to avoid this complication, a scientist called Punnett, an England dentist called Punnett, has given the Punnett square board. Okay, Punnett square board is represented like this. Here we write the male characters, and here male, this is female. Here we write the female characters, and then we draw a box like this. Okay, so this is Punnett square. Yes, this Punnett square may not be so useful in law of dominance, but in law of dihybrid cross may this Punnett square is very useful. Okay, now we are only dealing with monohybrid cross. Okay, this is called monohybrid cross. Why? Only one character we have taken, monohybrid. If you are taking two characters, then it's called dihybrid cross. That is different law. Okay, now crossing TT, you get capital T, capital T, small t, capital T, you get capital T, small t, small t, you get small t, small t, TT, 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 yeah, here capital T, sorry, TT, you get TT, TT, you get capital T, small t, small t, small t, small t, capital T. These I am writing here again. Okay, so capital T, capital T, capital T, small t, okay, and small t. So these are the three different characters that we have obtained, okay, when we have crossed F1 generation. And whatever these you have obtained, this is nothing but the F2 generation. Okay, just now I've said again, F2 generation, filial 2 generation, which is nothing but cross the progeny obtained when you cross F1 generation is F2 generation. Okay, now out of these three characters, we have to notice that capital T is only one. Small t are two. Small t is one. Okay. This is one thing. Next, I am writing this is genotypic. If you observe, I have written the alphabets. It is genotypic. Now I am writing this side tall and dwarf. Can I say this is phenotypic? Yes, this is phenotypic. Now, now you say me, how many are tall, how many are dwarf? Now I said tall gene is dominant. If you remember in that table, we have taken tall as dominant. Okay, so tall is dominant means this tall character here it's purely tall, no problem. Here this capital T is dominating small t, so even this is phenotypically tall. Genotypically, it's not pure tall. It's a combination of capital T and small t. But phenotypically, this capital T is dominating this small t, so even this and this both are tall. And this alone because both are recessive genes, no one is dominating another, both are dwarf. Okay, very clear. So tall is how many? 2 plus 1, 3. Dwarf is 1. Once again, I'm explaining. 1 is purely tall, capital T, capital T. 2 are combination of capital T and small t. And 1 is small t, small t. Genotypically, all are not same, but phenotypically, due to the presence of this capital T, this is dominating this small t, hence even this is considered as tall. So 2 plus 1, 3 talls, 1 dwarf. So 3 talls, 1 dwarf. From these two, I can come to one conclusion. What is that is phenotypic, genotypic ratios. Okay, phenotypic ratio, genotypic ratio. Now, what is phenotypic ratio? You say me now. Phenotypic means this one or this one, this one. Correct? Why? Physical characters. 3 tall, 1 dwarf. So can I say 3:1 is the phenotypic character? Yes. Now coming to genotypic. Can I say this is genotypic? So in genotypic, we are not taking tall and dwarf. We are only taking these into consideration. So can I say one is pure tall, one is hybrid, capital T small t are 2, and one is purely dwarf. So can I say this is 1:2, 2:1 ratio, can I say? So this is the phenotypic ratio and the genotypic ratio that is obtained when we cross capital T and small t. Okay. Okay, now this is clear. This is nothing but law of dominance. Okay, now you may try taking another two characters, that is maybe round and wrinkled, or maybe green and yellow. Okay, that you can try afterwards. Now let us you know talk about another case in law of dominance. That is just a second. Yes, that is test cross. Now we got in F2 generation that capital T, capital T, capital T, small t, small t, small t. And we are saying this is dominant, this is, a combination, a hybrid, and this is recessive. Now, to know, to know whatever is obtained, is it dominant or recessive, or is a hybrid, we got one technique, that is test cross. Okay, Mendel, when he obtained, the characters, to understand or to get the idea whether it is a dominant or a recessive one, he did the test cross. Okay, what is this test cross? Let us see. Test cross is crossing over, okay, crossing over of the unknown character with a recessive parent. Okay, with a recessive parent. Crossing over of the unknown character with a recessive parent. Okay, how you will get this? Okay, if you see in NCRT, 12th class NCRT, he has given the example of the flower color to explain test cross. Okay, we will take some other character. Why only flower? Okay, I am taking, suppose, First, I'm taking the recessive parent. Okay. Okay, let me say small t, small t, small t, small t. Okay. Dwarf and dwarf, dwarf and dwarf, two characters I have taken. Okay. Unknown character, maybe it can be capital T, or it can be capital T, small t. Correct? Let me organize it. It may be capital T, small t, or capital T, capital T. To know whether it is this or this, we are crossing it with the recessive parent. This is the recessive parent. This is the progeny that is obtained. Okay. So can I say capital T, small t, capital T, small t, small t, small t, small t, small t, capital T, small t, capital T, small t, capital T, small t, capital T, small t. Okay. So we have got this progeny here. Okay. Now next, just a second. Yes. So this is obtained. Okay. So here you have got both capital T, small t, small t, small t, small t, small t. And here, if you observe, all you have got hybrid. We can say if you are getting all hybrids, then the obtained is purely tall. Or if you are getting a hybrid here, or if you are getting, like the F2 generation case, or two hybrids and two dwarfs, then it is the capital T, small t that you have obtained in the progeny. Okay, this is called test cross, or also called as a back cross. Okay, next. Let us check for the another case in law of dominance. Just a second. Okay. Let us check for codominance, codominance, and incomplete dominance. Codominance, incomplete dominance. These are the two cases, of law of dominance. After completing these two, we can wrap up law of dominance. If you complete these two cases, law of dominance is completed. Okay. Codominance. The best example for codominance is blood group. Blood group, that is AB, O blood groups. So how AB, this thing is becoming the case of codominance. Okay, so why we say ABO blood group is codominance is, okay, we represent A as IA. It synthesizes A sugars. IB, IB, IA, IB. This II. O blood group doesn't synthesize any sugars. No sugars. Okay. This thing is there. Now, let me take these two characters. Okay. A, B. Suppose say a parent has IA blood group, pure IA blood group. Another parent has IB blood group, IB blood group. When these two are crossed, let me say this is male and this is female, and these two are crossed. So can I say whatever progeny is obtained, IAB, 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 IAB. AB is not the character of mother nor father, but it is expressed. So here AB genes are dominant genes. Okay, we have to know this. AB genes are dominant genes, while O is the recessive gene. Okay, recessive gene. That's why in the presence of A and B, neither A can dominate B nor B can dominate A. That's why both of them are expressed. That's why this is called codominance. Means neither one is dominating another. Both are expressed equally. But if suppose I am taking another case of, let me say, I am changing the male parent from let me say he's having IA and IO blood group. Yes. Now when we have crossed, I get IAB. The child may have IAB. The child may just be IB. O is recessive. O can't express itself. Or maybe he can be IAB, or he can be just IB. Okay. This is one case. Now if another I'm taking IB as, okay, small I, if I'm taking, okay, just a second. Yes. Okay. This is one case. Now, if I'm again changing this thing, okay, if I'm taking I, just I, okay, now this thing I'm crossing again. I am getting here IAB, IB, IA, I. Now, if you observe here, let me say, because I or O group is recessive, O and O are both are recessive. So both are expressed here. They are in homozygous condition. That's why they are expressed here. These things, these are already dominants. So here A is dominating O group, so only A is expressed. Here, B is dominating and only B is expressed. Okay. Now here the ratios will differ. The phenotypic and the genotypic ratios will differ. Okay. There is one table which we get for this IAB crosses. Let me try to draw that table for you. Okay. Okay. Let me say I am taking, I have B, AB, O blood groups. Okay, these are the four different blood groups that we are having. So can I say phenotypically there are four phenotypes, can I say? Okay. Next, genotypically, if I'm saying IA is one, IA, IB is one, I can say even IA is one. Okay. Next, IB is one. Next, IO, just one. IA, IO is another one. IB, or let me represent I and I is another one. Okay, even I can say here I and IA, or I and IB. Leaving these apart, these are the combinations that we can get. Only observe these. Okay, these are genotypes. These are genotypes. This is phenotype. So can I say there are four phenotypes and 6 genotypes, can I say? Okay. There are four phenotypes, 6 genotypes. This is about the case of codominance. Okay. Now this is done. Coming to incomplete dominance. So for incomplete dominance, we are having two cases. One is the snapdragon case, another one is the starch synthesis. Okay. Coming to snapdragon case. Okay. Uh, evening primrose also called as. Okay. In this, we will be talking about the flower color of the flower. Okay. In snapdragon, or also called as the evening primrose, snapdragon, let me say, dominant, recessive, dominant and recessive. Dominant may, we are having the dominant color as red. This is not pea plant, correct? In pea plant flower color we had violet and white, but this is not pea plant. Okay, we are taking here flower color. Okay, flower color. Dominant is red, recessive is white. Okay, this I'm representing it as capital R, this as small r. Take two combinations. Here I am taking let male be capital R, correct? And let female be small r, correct? Red, complete white. When the cross is done, you get here capital R smaller, capital R smaller, capital R smaller, capital R smaller. Correct? Now you must be saying, according to the concept that we have learned, that this must be red color because red is dominant, white is recessive. So red is dominating white, so red is expressed. But a small twist what we have is the obtained is pink color. The flower obtained is pink color. So that's why this is called incomplete dominance. Why it's called incomplete dominance? Because both red and white are expressed. No, both red and white, both of them are not expressed. Another different color is entering into the picture. Pink, nowhere pink is there. Dominant is red, recessive is white. We got pink from somewhere. That's why it's called incomplete dominance. Now, I'm rubbing this part. Whatever this RR, whatever the F1 generation pink color we have obtained, let me cross those two. Capital R, small r, capital R, small r. Correct? This is male, this is female. Okay. So whatever you obtain here, can I say capital R, capital R? Can I say capital R, small r, capital R, small r, small r, small r? Correct? Okay. Phenotypic genotypic ratio. Let me write the first phenotypic, next the genotypic ratios. Now, as I said, the obtained progeny in F1, these two were pink, these two were pink. This will be red, of course. Capital R, red pakka. Small r, white pakka. These two are pink. Capital R, smaller are again pink in color. So, which is explaining the incomplete dominance. Now, phenotypic ratio. Wait a second. Yeah. Phenotypic ratio is, how many phenotypes are there? One red, white, one, two pinks. Correct? So can I say 1:2, 2:1? Can I say phenotypic ratio? One red, two pink, one white. Genotypic ratio. Okay, one second. Genotypic ratio. Can I say same? One is capital R, two are capital R, small r. One is small r, small r. Now there is one deviation from Mendel's law. What is that? In both these two cases, in Mendel's cross, we got the phenotypic ratio as 3:1. But in both these two cases, we are not getting the same ratio 3:1. The ratio is getting deviated from 3:1. So here that is 1:2:1. That's it. Next case, last case that is left over in law of dominance is the starch synthesis. Two characters, capital B, small b, small b. Starch synthesis. Whatever this capital B is there, it is having, it is big round in shape and it's having more capacity for starch synthesis. This is opposite to capital B, capital B. And when these two are crossed, you are getting capital B, small b, which is having the intermediate characters. So this is having the intermediate characters. What do you mean by intermediate characters? This is neither completely synthesizing, exactly how this is synthesizing starch, nor it's completely round in color. It is having the characters of both this and this, but neither of them are expressed. That's the starch synthesis case. Okay, capital B, small b. Okay, so these are the different cases that one can come across in the law of dominance. Okay, all the cases I have done with, for small assessment like kind of thing. The link for that PDF is given in the comment box. If you want, you can go through that. The answers for that also will be uploaded shortly in my videos. Okay, thank you everyone, hope you enjoyed my video.